Guys, how's everybody doing today? I hope you had a good weekend. I apologize because you get to hear me sounding like this. I actually had to go in and do a COVID test yesterday. That was just so much fun. And now I get to wait for a couple of days to see what the results are. So I hope that you are healthy, and I'm going to keep this lecture pretty short for today. Before I start, I need to tell you, though, that there is a discussion that goes with this lecture. And again, this is one of those participation point discussions, and would also be beneficial to helping you remember the different organelles in the cell. So if you would go take a look at that, I've left it open until Friday of this week for you to post. And it doesn't have to be something crazy hard, just something that will help you to remember the different functions of the organelles. Okay, so this is chapter 4, and I'm going to start with my whiteboard here. Okay. So, chapter 4, cells. So I want to start, as usual, with some vocabulary. Let's talk about something called cell theory. This basically says that all living things are composed of cells, and that all cells come from other cells, okay? Okay. So cells give rise to other cells. When it comes to cells, there's two different categories. We have prokaryotes. It's an R. I know my R's look like V's. And then we have eukaryotes. Okay, so let's talk about the different characteristics of these. My lines apparently are affected by whether I feel good or not, because look at those. Those are the worst lines ever. Anyway, when it comes to prokaryotes, we're thinking about usually single-celled organisms. So things like bacteria. Okay. They are usually smaller in size. They lack almost all organelles. And I'll show you the one organelle they do have. They do not have a nucleus. And they tend to be a little less complex in structure. Okay. Eukaryotes, on the other hand, are going to be multicellular organisms, almost exclusively. You guys, that's hilarious. You notice I just made it worse. It's just... so sorry about that. So usually multicellular. So the cells in our body are eukaryotic cells because we are eukaryotic organisms. Okay? These tend to be larger cells and have many organelles. Many, many. They have a nucleus where DNA is stored and have a more complex structure. So yes, you need to know these things. You need to be able to tell the difference between prokaryotes and eukaryotes. Honestly, if you learn the characteristics of one, you know them both because they're just opposites of each other. Okay? All right. Now, let's talk about 4 things that all cells have in common. So whether they are prokaryotic cells or eukaryotic cells, all cells have these things. Okay. So first, all have at least one chromosome. So chromosomes, if you remember, are where genetic information or DNA is stored. And in order for those cells to make other cells of the same kind, they have to have genetic information. They have to have the instructions for how to do that. So even the most simple prokaryotic cell will have at least one chromosome. And then chromosomes code for proteins, and proteins are what create the structures in all living organisms. So in order to build proteins, that's a weird R, all cells have ribosomes. And again, this is true whether it's prokaryotic or eukaryotic, they all have ribosomes. When it comes to prokaryotic organism, ribosomes are the only organelle that they have. Okay, they also all have cytoplasm, that jelly-like fluid on the inside. I'll give you that definition in a minute. And they also all have a plasma membrane, which we'll also talk about, but this is essentially the membrane that surrounds the cell. Okay? So all cells have these things in common. Okay? Remember, it's both prokaryotic and eukaryotic. Or prokaryotes and eukaryotes. This is just a different version of the word. When it comes to these prefixes, prokaryotic, this is karyo. This is referring to like before the nucleus. And eukaryotic, U is true. So in other words, before the nucleus and having a true nucleus. Okay. Switch over here. So let's do a couple of definitions. All right, so I've been talking about organelles. What are they? Organelles are simply membrane-bound or membrane-enclosed structures that perform a specific function in a cell. Okay, so you have the cell, which is enclosed in a membrane, and then inside that cell, you have a bunch of smaller little structures that also are surrounded by a membrane. I'm going to show you real quick. This PowerPoint slide is available under modules so that you have access to it. All right, so what you're seeing here is an animal cell, or actually several animal cells. And you can see the plasma membrane, which is this. And I know that these have kind of a cube or rectangular shape. You might want to think that these are actually plant cells. These are just a type of columnar cell, skin cells more than likely, attached to a basement membrane here. Anyway, you can see the plasma membrane, and then this large organelle, this is the nucleus. So you can see that in each one of these. What you can't really see are the other organelles. The nucleus is by far the largest. You can identify it under a microscope. But trying to look at the other organelles under a microscope is, at least by a regular light microscope, like the kind you find upstairs in the lab in the science building, that's not going to happen. You have to have something that's more sophisticated, like an electron microscope, transmission electron microscope, something like that. Okay, let's go to the next, if I can here. Reason it doesn't want to go. Okay. So this is another look at cells. This one, these are actually cheek cells. I believe that you guys still do in lab, if you're in 1015, you take a swab of your own cheek cells using a toothpick, and then you stain them. This blue color is a dye that's used, and you put them under a microscope and you can actually see them. And this is exactly what they look like. So these are epithelial cells, or the cells that are on the top layer of skin. This is the plasma membrane. And this is the plasma membrane of another one. There's another one. And then these here are a bunch, all kind of jumbled on top of each other. The reason I like this slide is because it gives you a look at the plasma membrane itself and shows you that it's not rigid or stiff. It can adapt and bend a little bit, so to speak, depending on its environment. So it's not rigid like what you'd find in a plant cell. And that's because they don't have cell walls. Only plant cells have cell walls. But animal cells do not, and so they are somewhat flexible. Ooh, made that huge. Okay. So this is an animal cell. And like I told you, we have this plasma membrane right here. And that's really referring to all of this. Okay. So all of this out here is the plasma membrane. And it goes around the cell. Okay. So I won't draw. Well, I guess I will. Plasma membrane. Then inside we have all of these different organelles, right? So that's an organelle, and this, and this. And these guys are all surrounded by their own membrane. So it kind of keeps them separate from each other while all being in the same system. You might notice the word organelle sounds a lot like the term organs that we use in our larger anatomy. It's because they mean like little organs. If you're having a hard time picturing all of this with a cell, you can think about the body in general. Inside of our body, which is covered in skin, the membrane essentially, we have a bunch of different organs. Respiratory tract, the lungs, bronchial, trachea, all of that. And then we have digestive tract and the circulatory system, and we have all of these different organs and organ systems all packed within our bodies. This is how cells work. You have the larger cell itself with a bunch of these smaller organs, organelles, that each have their own function inside the cell itself. Okay. Let me come back over here to my whiteboard. Okay, so let's go over some of these organelles. I'm actually going to start with ribosomes. So ribosomes are responsible for protein synthesis. Okay, in other words, they're the ones that are actually going to put proteins together. If you remember from our chapter last week about macromolecules, we talked about amino acids being monomers for proteins. So it's the ribosomes that string these amino acids together to build a larger protein. And again, without a ribosome, or without ribosomes, plural, cells aren't able to make proteins, which means that there really wouldn't be living beings or structures without them. Everything from a lot of the different hormones and organs in our body are built of proteins. Anything that gives you a trait, your hair color, your eye color, skin color, all of these things are proteins. So we have to have ribosomes. Then we have something called the endoplasmic reticulum. I love that word, right? I always abbreviate it the ER, kind of for obvious reasons. You don't want to type that whole thing out. So if I were comparing a cell and its organelles to a functioning factory, the ribosomes would be responsible probably for building the machinery in the factory. Then we have the endoplasmic reticulum, which is kind of the main manufacturing facility as far as making cellular products. In other words, the endoplasmic reticulum is responsible for making lipids, which include our steroids and hormones. It's responsible for making more cell membrane. It's responsible for making components of proteins that will go into a finished functioning protein. Okay, so it's kind of like your main manufacturing facility. So if this is an actual factory, the endoplasmic, let's say our factory makes teddy bears. The endoplasmic reticulum is going to be the part where those teddy bears are assembled. And what makes the equipment for the assembly are the ribosomes. Okay, so there's two types of endoplasmic reticulum. We have smooth ER. Smooth ER lacks ribosomes. Okay, no ribosomes And they are again used in lipid synthesis, so for steroids, hormones. Um, okay. Then we have rough. What makes rough ER rough, you ask? Ribosomes. Okay. Um, and again, this rough ER is going to be used, uh, as you might suspect, in protein assembly because it has ribosomes. What are ribosomes for? Protein synthesis. Okay. So what it means is when it comes to ribosomes, I'm going to add a little note up here. Ribosomes can actually be found either free in the cytoplasm, just free floating, if you will, in the cytoplasm, or they can be found embedded in the endoplasmic reticulum, which is what makes rough ER. Okay. Regardless of where they are, their function is the same. So it doesn't matter if they're in the cytoplasm or if they're attached to the rough ER. They have the same job, which is protein synthesis. Okay. All right, then we have the Golgi apparatus, which is named after a person. Sometimes you'll also see this called the Golgi body. Same thing. So the Golgi apparatus works with both types of ER. And again, if this was a factory, I always think about the Golgi as being like the warehouse facility. Because the Golgi's responsibilities are to receive, find, store, and distribute cellular products. So in other words, we have our assembly line or our manufacturing facility with the ER, and it's building its proteins, it's building its lipids, and it's building cell wall and all of these different products, some of which are going to be used inside the cell, some of which are going to be shipped outside of the cell. Well, after the ER finishes with their manufacturing, it sends the product that it's been manufacturing to the Golgi. And the Golgi then is going to receive it. Again, think of a warehouse, refine it. There are sometimes some finishing touches that need to happen that the Golgi can complete. Store it. So think about the warehouses, having pallets of products, and then distributes it. The trucks will come and pick the products up from the warehouse. So this is what the Golgi does. It's obviously an extremely important function in the volume of cellular product that it comes in contact with. We have our lysosomes. Lysosomes can be like your trash compactor or your furnace, because this is going to break down harmful wastes or foreign substances. So the cell's going to have leftovers sometimes of things that it doesn't need. Old organelles will be broken down and destroyed and replaced with new ones. All of that gets sent to the lysosomes. The lysosomes have digestive enzymes, which will tear those wastes or foreign invaders, so like virus particles, bacteria, and stuff like that. We'll tear them down, break them apart. You really only find lysosomes in animal cells, and they are an extremely important part of the cell. There are some genetic disorders that have to do with lysosome deficiencies, where the cells are not able to break down waste, and they can be deadly. What else is I going to tell you about this? Oh, obviously, we're going to want to keep the digestive enzymes within this membrane, much like you want to keep all of your digestive juices in your stomach, because when it gets out, it starts eating through all of the other tissues in your body. Well, when lysosome digestive enzyme gets out into the rest of the cell, it will digest the rest of the cell. Lysosomes then also play an important role in something called apoptosis, or programmed cell death. If a lysosome finds that, or I say lysosome, if the cell determines that it's infected with something, bacteria, virus, or that it's cancerous, it will signal the self-destruct process, which involves puncturing holes in the lysosome for those digestive enzymes to leak out and consume the cell. Get through the last of these. Let's talk about mitochondria. So mitochondria, as I'm sure you may recall from your high school classes, are the powerhouse of the cell. This is true. And again, if we're talking about a factory, then the mitochondria would be your power plant. This is the location of your cellular respiration, which we'll talk about in chapter 6. Cellular respiration produces energy in the form of something called ATP. You don't need to know all the details about ATP right now because you're going to learn them in chapter 5. But just know that this is where it happens, and this is why they're the powerhouse. Cellular energy is made here. The interesting thing about mitochondria is that it has some of its own internal structures. So first off, it has, it's surrounded by a membrane, and inside that membrane, we have something called cristae, which are kind of this, what do I call those? And then it has something called the matrix. No, not that matrix. The matrix is similar to the cytoplasm, and that is a thick jelly-like substance. I will show you a picture of that in just a second. Then we have the nucleus. The nucleus is the brain, if you will. This is where the genetic code is held. So in our factory, perhaps this is where all of the software is stored with all of the different software programming needed to run this factory effectively and to build new factories just like it when needed. So it contains most of the cell's DNA. Yes, most. Because the mitochondria itself also has a small mitochondrial DNA is inherited through the maternal side, so you have to link it through living maternal relatives. It's become an important part of genealogy, and also can be used in forensics. It has some different information than what we keep in the nuclear DNA. Contains most of the cell's DNA. It also has a double membrane that we call the nuclear envelope. The nuclear envelope has pores, called nuclear pores. And it's through these pores that genetic information and cellular products can come in and out of the nucleus. The nucleus is heavily guarded, if you will, with these double membranes. So the pores are going to allow for stuff to get in and out. It also contains something called chromatin. Chromatin is just, how do I want to put this? It's uncondensed chromosomes. So in other words, it's a mix of protein. That X-shaped chromosome structure that we're used to seeing when we talk about genetics, that only exists when cells are dividing. Otherwise, it's unwound or uncondensed in the cell, in the nucleus specifically. Finally, within the nucleus is something called the nucleolus. The nucleolus is crazy important because this is where ribosome components are made. Okay. So when it comes to ribosome components, the nucleolus makes them. The components are taken out into the rest of the nucleus, out through the nuclear pores, and into the endoplasmic reticulum where they are finished. Once they're finished, they either go hang out in the cytoplasm or they're attached to endoplasmic reticulum to make rough ER. Yes, this is the thick. Don't confuse cytoplasm with matrix. They are similar in composition, but they are distinct and they are in two different places. One way you can remember it is that cytoplasm starts with the C for the cell, and matrix starts with an M for the mitochondria. Okay? So that's how you keep those terms from being confused. Let me go ahead and switch real quick. Okay. So last thing I want to show you real quick again. All right, so I already showed you where the nuclear envelope, or I'm sorry, where the cell membrane was in this. This is the outside part, okay? Let's look at the nucleus. So here's our nuclear envelope. Obviously this has been cut in half, or into quarters, if you will, so you can kind of see. Nuclear envelope. And within the nuclear envelope are all of the nuclear pores that allow substances to get in and out, including ribosome components. So here are all these pores. Okay. And then you'll notice here, this pore is showing its connection to the endoplasmic reticulum, which would happen in more than one location. They're just showing it here in one. We've got the nucleolus, where those ribosome components are made, pass through the rest of the nucleus and out to the endoplasmic reticulum. All of these little squigglies in here are supposed to be chromatin, so that unwound DNA and protein that's in here. Okay, when it comes to our endoplasmic reticulum, we have this kind, which as you can see is studded with all of these little tiny ribosomes, so that we know this is rough ER. So is this. Then we have this right here, which has no ribosomes, and that is smooth ER. Remember that they just have two slightly different functions. And then we've got more ribosomes that are floating free in the nucleus, or I'm sorry, in the cytoplasm. We have our Golgi apparatus, which again is the warehouse, receives, refines, stores, ships. We've got our lysosomes. These are the digestive sacs of enzymes, or sacs of digestive enzymes, right? That are going to destroy foreign invaders. And then we have, actually, let me pick a different color here. We have our mitochondria. Powerhouse. You know, this image makes it look like you only got a couple of organelles in a cell, but that's not true. There's dozens and dozens. Some types of cells have more mitochondria than others. Some have more lysosomes than others. Some have more endoplasmic reticulum than others. It just depends on the cell and its functions. I'm going to skip past the plant cell part because I'm not really going to go over that anymore in this lecture. Mitochondria. I'm going to talk more in depth about mitochondria when we get to cellular respiration. For now, I'll point out the double membrane system. And then we've got the inner membrane called the cristae right here. So all of these foldings, how it folds. This structure of the folding back and forth is extremely important for ATP production. But again, I'll talk about that more when we get into cellular respiration. And then, of course, inside is our matrix, that fluid-like, that jelly-like fluid that's inside. Okay, also going to be extremely important in cellular respiration. All right, guys, that's all I got in me for today. I've given you all the information you need to start on your discussion. So you can go ahead and do that with what I've given you. Remember that you cannot use a factory as an example because we've used it in our lecture instead. Okay, I will have the rest of this lecture up for you by Wednesday. Thanks, guys, and stay healthy.